So anyway, the blog that's coming out about mycotoxins this month, one of the things when I was talking to her about it was about milk. So, we do a lot of the yogurts and the dairy, but some people have issues with it. And the dairy cows in particular, if you don't know the source of your milk, if you're using a big pooled milk provider like the big names instead of a local dairy or something, especially in the wintertime, they are fed silage. And that corn silage can have a lot of mold and mold toxins in it. And then the cow eats the silage and the cow has the toxins. So that was something I really I don't know why I hadn't thought about that, but you are what you eat. And, mothers know when they eat things that disrupt their baby that they're breastfeeding digestion, you know that. So it makes a lot of sense. So I started wondering if, maybe it's better to make the yogurt when the grass is green outside and maybe not so much in the wintertime when they're being fed other feed. So I don't know if anybody has a thought or a comment on that, but I saw Debbie smile. So
am I on?
Yes, you are.
No, I was just saying I make coconut milk, but right now, I'm out here in Wyoming, and so they're harvesting all the hay, and they keep doing all these different cuts of hay and stuff. So I don't know if all cows get fed silage in the winter, or if some of them are getting the hay that I'm seeing like in these ginormous bales.
So the beef cattle generally get the hay.
Okay.
And I would guess dairy cattle maybe get some hay. I mean, I shouldn't say that because I don't know dairy cattle that well, but I do know that a couple of people that we've worked with on some research projects, specifically work on silage, corn silage.
Mm-hmm.
And, we did work with our glyphosate remediation stuff in corn silage.
Mm-hmm.
And which is interesting too, because Dr. Don Huber, who's the kind of one of the top experts on glyphosate, said we're having a lot more fusarium now in the corn and the soy, and both of those, soy cakes and corn silage, are fed to the dairy cattle, and glyphosate selects for fusarium. So we're getting more and more fusarium because it's resistant to glyphosate where other organisms are not. So I found that pretty interesting. And I don't know if anybody watched this past week, Children's Health Defense had a online program about kind of what's going on in the food, and they had Dr. Huber on there. Let me see if I can actually, it may still be viewable. I don't know how long it's going to be viewable, but it was called The Attack on Food. So I think maybe you can still watch it. So I'll post that here in the
Oh, wow. Thanks.
Yeah.
chat.
I found it on Twitter.
And they had Stephanie Seneff talk. I still haven't had a chance to watch her talk because I had something else going on when she was talking. But Don Huber gave a talk, and he actually talks about a little bit about our cabbage study and the Paleo Power, which is Yield and Shield on our website, but about the remediation of glyphosate with that. So it was a pretty good lineup, just talking about what's going on in the food supply and what you can do about it and how to be more proactive with your local farm community, and trying to get us away from this major industrial food supply. Yes, Elizabeth.
Hi, I don't know if you saw, there was an article today in a scientific publication that bats died. There was a big bat die-off. So the bats eat the mosquitoes. So there were more mosquitoes. So the farmers, and this is in the States, use more pesticides. And there was a lot of children mortality. Did you hear about that?
I did not hear about that.
I have it. New Scientific, bat die-off led to more insecticide use and more infants dead in the U.S. 1,300 deaths in children under 1 year old. Anyhow, that's the.
Well, I would say that data would need a lot more digging into.
Yeah, but
because a lot of times
course.
those headlines are kind of designed to send you off on the wrong trail. But I will say there is actually a book called The Moth and the Iron Lung. I don't know if you're familiar with this, but that's about polio and the use of Paris green, which was an insecticide that was used against the gypsy moth. And the story actually follows the movement of what was called polio, with the with the migration of the spread of the gypsy moth and them using this very toxic chemical.
Wow.
And so, sometimes there's like finger pointing away from the actual culprit, and the truth lies somewhere a little. But that's very interesting. I'll have to look that. that up.
Yeah, it was in the New York Times this morning, which doesn't mean anything. Just where to find it. But well, I mean, using more insecticide would clearly affect
Right.
humans, right?
Yeah, right.
And children are vulnerable.
So also the one of the things that I don't know if Dawn actually talks about it in this Door to Freedom series or not, but one of the things that we discussed when we were working on the publication about the soggy cabbage and the glyphosate in the poultry was that during COVID, because there was a labor shortage of farm labor, so in 2020 and 2021, the farmers used more glyphosate as a drying agent in their crops because they didn't have, I mean, I don't know if that actually makes sense to me, but they didn't have the same amount of labor. And so they basically sprayed the crops or they sprayed the crops more
Yeah.
to dry them faster.
Well, it helps them. It helps crack the wheat.
But then does it make it more stable until they can harvest it? Maybe if it was taking longer to harvest.
Yeah, so I'm not really sure. But you know what's also interesting, because they may have used more on the wheat. So wheat produces shikimic acid, which is an ingredient in Tamiflu. And wheat will produce more shikimic acid when glyphosate is put on it. I stumbled across this 4 or 5 years ago, which I thought was a very bizarre connection. But if you think about Tamiflu, I don't know if anybody was given Tamiflu during COVID or not, but that just seemed like a sort of a weird connection to me. But But what happened was the poultry manure from 2020 and 2021 had a much higher parts per million of glyphosate in it than 2022 and 2023. So, I don't know if that is in part because the poultry producers kind of figure out maybe it causes more death in the birds, but nobody's going to be talking about that out in public. But so, and I have a article coming out about that research we did with Dr. Huber. If you are a member of Weston Price, I think most of their stuff is online like a little bit later if you're not a member, but if you're a member, they do a printed journal every quarter. And the one that's coming out, I guess at the end of this month, actually has an article about the soggy cabbage and the work with Dr. Huber looking at the poultry manure because it's used on organic crops. So you can use conventional manure on organic crops. And this is kind of a dirty little secret that organic farmers are not really talking about. And it's also interesting because I had a call 2 weeks ago with a guy who owns a ag seed company. So he sells seeds, and he sells from organic all the way up through the, Roundup Ready. He's got the whole lineup. But he's gotten very focused. He's an agronomist, and but he also has a background in I think his master's or PhD was in weed management, which is, basically trained in pesticide or herbicide use. And he heard Dr. Huber speak a few times, and he actually told me the last time he heard him speak, he said, what was the word he used? I was convicted by the Holy Spirit. And, he said, I know it's very dangerous to talk about this, but he's starting to look at it, and he has fields that he does testing of different seeds and what the yields are, and, work where he's trying different things. And he didn't think he would have that much glyphosate in his soil because, of course, he's been educated that it doesn't stay in the soil. And he tested his soil, and he was just flabbergasted to find that he had 1,100 parts per million, or parts per billion. I can't remember if it was parts per billion. But so the limit, Dr. Huber says at 500 parts per, I don't know if it's million or billion, but you start to have an impact on the nutritional content of the food because of the chelation of the trace minerals. And so, having that conversation with him, I was also talking about the Amish farmers because he has a lot of Amish. And I have quite a few Amish customers who take Sugar Shift through a healthcare practitioner who works in the Amish community, and they have a lot of health problems. But as it turns out, the Amish have been using glyphosate and other herbicides in their farming operations. And I think there's a tendency for people to think that they're all natural, but they're not. And it's actually worse for them because, the farmers who are in the big operations, they're in enclosed tractors where they're not getting exposed to it. But these Amish farmers are actually, they're on a standing, like buggy type thing right behind the horses, and the sprayer is right there. So they're constantly exposed to that. And, and they don't have protective gear on.
And they also stay within their community in terms of trading. Like, so if they're all using those chemicals, then they're all, essentially contracting what their neighbors are using as well from the foods they're eating.
Right. So, anyway, it's an area of focus for me because I work with he's a brilliant healthcare practitioner and he knows glyphosate is an issue. I don't know that he knows, how much of it is actually used in the farming operations there.
Martha, would you mind saying really quick what it is that Sugar Shift does that kind of neutralizes glyphosate and helps essentially detox it from the body?
Let me see if I can find this.
Because that's right?
Right. Yep. I'm pulling up something here. Okay. Let's see. I have to go to this database, I think. Now it's interesting. It's not coming up now. Let's see. Oh, wait. No. Pathway.
I have another quick question. Sorry, when you've got a chance.
Sure.
So like I mentioned, I am just reading Super Gut, so being educated on all that. And at some point, I'm curious to know because your probiotic isn't included in his original because he didn't really know about it, or I'm not sure if it was even developed then. But I think now he really
it's in the book. It's just mentioned in the back.
Oh, okay. So I'm not quite there then. But I know he kind of exclusively uses or utilizes Sugar Shift now, doesn't he? He really likes using that above all. You mentioned last time on the last call that you weren't sure about the yogurt piece of it or doing it for as long, cooking it for as long, and the temperature and all that stuff. I'm just curious to know. I know at one point, the point I'm at in the book, he talks about the hydrogen levels being higher when you do El Reuteri yogurt. So you have to be kind of conscientious about that if you're testing your hydrogen or whatever hydrogen levels on your breath. So do you have any thoughts about that? I'm just curious.
Repeat the second question
about the Sugar Shift he originally talks about El Reuteri yogurt and then talks about using, like the BioGuy or whatever. And
right.
But he talks about at one point the methane levels when you're testing your breath, especially in the early stages, that if you test your breath,
it can be right. It can be higher.
Yes.
I'll say I'm definitely not the expert on his breath testing and that, but what I will say, with the single Reuteri or the Reuteri Gasseri approach, I think he does talk about methane and hydrogen sulfide. I think it's the hydrogen sulfide maybe more than the methane, but Sugar Shift, because it's like a balanced working system. You don't ever get too much of anything because it balances. They're sort of feeding each other. So something else will use that hydrogen before it gets too high.
I think that would be a good test with the air device. But Colleen, is your question that so you're treating SIBO, I presume, and that's why you'd be using your air device. If you're using the SIBO yogurts, then your air device, you've got to just put it away for a while. It's not going to be really accurate.
Yeah, I think that's so.
But if you're not treating SIBO and you want to take the sugar shift and you want to work on your metabolism, are you asking if the air device is accurate with sugar shift?
Yeah, I'm asking actually if Sugar Shift would have the same results, or if anybody knows based on experience, if Sugar Shift would have the same, Sugar Shift yogurt would have the same results as, say, the BioGaia Gastrus or whatever, that's just the single, L. reuteri, which I think you answered, because it sounds like if yogurt is made with, which I have made with the Sugar Shift, but of course I don't have the testing mechanism to know what the balance of the microbes are. But if you make it with the Sugar Shift, then it's a more balanced yogurt. It's not specifically just L. reuteri that sort of prevails, right?
Right. It is.
Mhm.
You know what's really interesting? This pathway that our organism has was in the KEGG genome database, which you probably don't know what that is, but it's an NCBI database, and it's been removed,
What?
which I find kind of interesting. It's not in the database anymore. So I'm going to have to do. I don't like, in order for me to go dig around and find it. Like, I would have to stop
Yeah.
talking and
Yeah,
not
you
do the
could
phone call. So I can do that, but I mean, essentially, there's a CP lyase, which is a carbon phosphate lyase that breaks down glyphosate, and that produces AMPA. So AMPA is, I forget exactly the mouthful, but AMPA is actually more toxic than glyphosate.
Mm.
And so, like in the testing, when we test for soil remediation, we test that it gets rid of both glyphosate and AMPA.
Mhm.
And so there's CP lyase, and then there's a pathway called the sarcosine pathway, which I believe also produces some AMPA. And then the third pathway breaks it all the way down. And one of the byproducts of that is actually glycine. So there are no toxic compounds in the third pathway, but it's much more complex, the steps involved. So it requires a lot more genes. And our plantarum has that capability. So, and we're in the process right now of doing a second test. We're waiting for the data to come back, but Raul did another test to like show at different levels of glyphosate in the petri dish with the strain and with Sugar Shift to see if what the before and after is. So, but I'm gonna show you, hopefully this comes across. So you will see some of this is in Spanish. But I think this page I actually got the translations right. So, it's a symbiotic formula. It's 8 strains. They cross-feed each other. And we use this predictive model that we built to show how they cross-feed each other and how they work together. So our initial goal, I think I've talked about this quite a bit, was to produce mannitol because it had been shown to stop the aggregation of the proteins in Parkinson's. But, what we found along the way after, making this formula to convert glucose and fructose into mannitol was that it does so much more than we ever even imagined. So you can see this is our computational model, and I'll see if I can enlarge this. And they cross feed each other.
Mm.
So, I mean, in our model, this is just one of the examples of in the model. So we can put other substrates to see how they're working. But you can see here that the bifidobifidum is consuming the glucose. The Bacillus subtilis is consuming that hydrogen. So it keeps things in balance by, not having excess hydrogen. And the plantarum is consuming the acetaldehyde. And that's kind of interesting too, because there's some evidence of increased acetaldehyde in people with Parkinson's. And of course, I made it for John with his Parkinson's, but there's also another probiotic called Neurally. And it was formerly called Solus, that is a single strain of plantarum that they have studied in Parkinson's. And I wonder if it's not partly due to removing that acetaldehyde. So that's our model. And then this, we use the prediction, and these are in Spanish, I think, but you can see mannitol production, and you can see butyrate production by the group. And then this is butyrate by strain. So, some strains are butyrate producers and some are not so much. So, these are not butyrate producers, but these are butyrate producers. And then this, again, it's in Spanish.
Butyrate is like one of the keystones for develop, like within yourself, amino acids and that kind of thing, building blocks, right?
Well, and it's the primary food for the cells of the lining of the gut.
Okay. Yeah.
So if you're not producing butyrate, you're not feeding those cells in your gut. So this was part of the work we did for our patent, basically showing each of the strains can do a little bit of conversion of mannitol, and well, some of them can't do it at all, but leuconostoc and the plantarum were the bigger producers. Bacillus subtilis is a bit. Reuteri is supposed to be a mannitol producer, but I don't know which one of these numbered is Reuteri, but some of them did not produce anywhere near the amount of mannitol that the combined formula did. So, it demonstrated the synergism of the strains together to reduce more of that glucose and fructose. So, and this is just the timeline of our clinical study. And so you can see here the changes in sugar shift in the fasting blood glucose and the controls, which are red. This was postprandial blood glucose, which one thing we don't really know if they were doing these at the same time of day, the appointments, which, could have had a factor here in the middle, but this was the 2-hour postprandial. I guess, no, that wouldn't make sense. Anyway, at the 90-day point was where we started to see the change here, and you can see the increase. So the control group was actually getting higher blood glucose at the 90-day point, and ours was sort of a gradual decline, which I think is statistically significant. But we did go on to take this out, and we took some of these subjects, we unblinded at the end of the study and took some of the subjects another 90 days. So at 180 days, Raul doesn't have that data in here. There's a much more dramatic drop off in the fasting and the postprandial, but We also had, a shift in diversity. And I think that is free fatty acids, but this was really, I'll say, one of my babies of really wanting to look at this. So serum lipopolysaccharide, and if you've listened to Dr. Davis at all, he talks about endotoxin and how those SIBO bacteria are producing endotoxin. And the problem with endotoxin, I mean, it can be quite dangerous in sepsis. Endotoxin can be, and your immune system's reaction to it can be a cause of death. So what I had looked at was there were a number of animal models using this endotoxin lipopolysaccharide. So there are animal models for Parkinson's, there are animal models for diabetes, there are animal models for depression, and animal models for multiple sclerosis, all using basically endotoxin to induce the symptoms. And so we decided to measure endotoxin. It's unfortunately, like if you went to LabPOR or Quest today, it's not an easy test to get. I mean, they do it in the hospital when people are, have an infection, but it's not something that's readily available as a measurement tool for you to see what your endotoxin load is. So, but, we had a dramatic drop in endotoxin level. Now, they did also have some endotoxin reduction in the control group, which that group took basically all the fibers so the inulin and mannitol that are in the sugar shift formula and the cellulose, their capsule was basically everything except for the bacteria. So you can get some protection against endotoxin with different fibers. And I know some of the research now with pectin in Alzheimer's and stuff, they've been looking at that. So I'm gonna out of this. I don't know. Oh, I do have one more slide here. Nope. Go. I think. What do I do? Okay. So this is actually some of the enzymes that changed dramatically in the before and after of the microbiome. So this 4-keto reductase went up by almost 600%, which is that key enzyme in converting fructose to mannitol. So, it's very clear in the microbiome that it is doing what we designed it for. And then this particular enzyme that increased 70% actually addresses problems with iron and iron homeostasis. And one of the contributors to LPS is often pathogenic bacteria gaining access to the iron in our bodies. Iron feeds pathogens, and I will have Morley Robbins on a bit later, and he can talk a bit more about that. But the lack of iron homeostasis, and there's a lot of added I'll say synthetic iron, but added iron to the food supply now. If you look at most fortified grains and bread, because they basically have milled all the nutrition out of the food and then they put it back in artificially, and iron is one of those things. Is that me? Oh, yeah, sorry. Not gonna answer that, but And bacterioferritin also is helping to manage the transport and recycling of iron so that you don't get the iron that's causing oxidative stress. And then this acyl-CoA is a gene that's involved in lipid metabolism. And we did also see in our clinical measures that there was an improvement in triglycerides, which is interesting. So this is showing you from our microbiome data. The blue is day 84 and the red is day 1. And these are all these different lipids that are involved in LPS. And so you can see there's a dramatic drop off in every one of the lipids in the microbiome that are involved in synthesis of those endotoxins. So I think that, oh, and this is just the structure of those. Probably don't need to go into that because that's more of a bowl. Now let me get back over here and unshare.
So taking sugar shift then helps decrease the endotoxemia.
Yes, ma'am. Yes, it does, Mary.
Okay.
So that
good to know.
we were really thrilled with those results because that's also an indication that, you know, it can be beneficial in a lot of inflammatory situations by removing that endotoxin.
Lots of endotoxemia.
I have a quick question for you about yogurt possibilities. And I know the idea of that it's not just for sugar shift in particular. Do you know, has anyone studied or do you know sort of what the balance is in terms of, because I know, you know, he talks about different yogurts at different temperatures and different times. So what did you find is the most optimal for sugar shift in particular?
You know, so I'm going to tell you, we have not studied the scientific metabolites or anything related to how many CFUs, because every batch of yogurt you make is going to be different.
Right.
So, you know, I could go test some stuff. Doing single strains, I don't know why I'm freezing. When you're doing single strains, you can measure to some extent what that's going to be like, but every batch is going to be different. And, you know, I learned that also with our producer who grows our strains for us. But, you know, these microbes are individuals with environmental impacts, and, you know, it can be the barometric pressure or the outside temperature or, you know, something that was different in the milk. So from the perspective of, you know, trying to give somebody, you know, if you go this many hours, you're going to have this many bugs. You know, what I'll say is because it's a consortium that's working together, you're probably not going to get the same kind of like growth curve.
Mm-hmm.
And you know, you get a more, I'll say you get a more sustained
Yeah, you may not get the same. I mean, it might actually create a lack of balance.
If you go, I mean, if you go too long, yes, I would say. So, I mean, I usually watch mine, and I'll pull it anywhere from 24 to 36 hours. But I have pulled it earlier because it's still a nice consistency. So,
and it depends really on the milk product you use too, for sure.
So,
yeah.
What about juices? That's what I wanted to ask you. Have you grown the sugar shift in juices?
I have not done that, but I actually have a young woman who used to work for me, whose sister made kombucha, used it as a kombucha starter, which is kind of weird because it's not a kombucha starter. But anyway, she fermented tea and juice and stuff like that. I'm trying to think if I've ever done that. I don't. Well, I don't drink a lot of juice. I did get some of the like kefir juice starters from Cultured Food Life and try that, which I almost exploded my house with.
Oh, my goodness.
Like I blew the, the little lid off it.
Yeah.
Um, I don't advise
Yeah.
doing that. But so I'm probably not the best expert on juice, but I like when you have a nice community like that. So the sugar shift formula, while it wasn't based on fermented foods, as it turns out, it is a lot of strains that are found in fermented foods. So the like Leuconostoc, you don't find that in a lot of I have rarely found that in a commercial probiotic that you can buy besides ours, but it is found. Leuconostoc, plantarum, and Bacillus subtilis are the three organisms that are found in many, many, many fermented foods all around the globe. So they're not just, unique to, my backyard in Colorado or whatever. So
So the sugar shift would might be something you could add to your like fermenting vegetables, making the sauerkrauts and stuff like that.
oh yeah, so yes, you could definitely do that. And I know John, is it Stansmore? John, who's he comes to our book club. Sometimes he comes to these, but
Yeah.
he does the sweet potatoes.
Mhm.
He
With
does with
with-
sugar
with that.
shift.
Oh, he does.
Yeah.
It ate the what is that stuff called? Oxalates, but I don't think it eats oxalates. Right? Sweet potatoes are very high in oxalates.
Oh, they are? That's
Oh,
interesting.
yeah.
So I actually well, I've talked about the beet kvass. I have used a sugar shift capsule before in my beet kvass, but usually I don't add anything to it at all. I just
Another food high in oxalates.
but if you're making at least this is my theory. I have to look. I'm sure it's in the literature somewhere. But so if you make beet kvass, you are getting organisms in your fermentation that are tolerant of salt. They are tolerant of oxalates. That like they break that stuff down. So I kind of feel like the beet kvass is, potentially something that could help people who have issues with oxalates.
According to Sally Norton, fermentation does not decrease oxalates. So
Hmm.
I am trying to, I have a lot of oxalate stuff going on with my body.
Martha,
Over
oh.
these past years, I've just eaten too many high oxalate foods every single day, 3 times a day.
Oh, yeah.
So I'm trying to eliminate them from my diet, except for the ones I use to prevent
Right.
whatever that is. I can't remember what Sally Norton calls it when you have too many oxalates being dumped into your body, and trying to get rid of it.
Mary, carrots work pretty well. I'm not sure what the oxalate content. I don't
Carrots
like
high.
it at all, but cooked carrots, you just need to make sure there's enough carb in there for the microbes.
Yeah.
And it needs to be watered down a little bit. Sweet potatoes do ferment really nicely. It's a little baby food-ish, but it's, if you get tired of yogurt, it's a nice way to go.
I did a sweet potato before this. I did it last year, and I made it into a frozen dessert. It was
Nice.
delicious. I had 2 layers. One was the pumpkin, or rather sweet potato ferment, and then a yogurt on top of it, and then another blackberry fruit on top of that. It was delicious.
A parfait. That sounds really
Yeah,
good.
yeah. But yeah, like I said, I'm not doing the oxalate foods right now. And that was another thing. I've already lost weight so that I don't want to lose any more. I'm down to like I'm down really low. And so I'm
Are
not
you
even
losing
on
muscle mass?
I don't think so. I'm maintaining my exercise routine. So I don't think I'm losing muscle mass. And I'm eating
Okay.
high protein and a lower fat because of my gallbladder issues, lack of gallbladder.
Mhm.
But I'm losing weight still. So,
You know, I mean, this is kind of interesting because most people doing the Rooterod do lose weight, but they're very early on in one of the publications I read that Rooterod could actually help you. Well, you put on muscle mass, so you can
right, yeah.
gain weight with the muscle mass, I guess. But
It's hard for me to say that I'm pretty sure I'm not losing muscle mass because I've increased my walking now. I was down to only one block because I was having a lot of pain. But now I'm walking more than 1.5 miles every morning. And
well, that's great.
and I swim and do other things also. So but I've limited my carbs and everything, trying to get over all this garbage that's going on in my body. So
Mary,
Yeah.
it might be worth getting one of those like Withings brand or a similar smart scale, so you can really watch and make sure you're not losing muscle mass. They're not totally
have-
accurate. You have one? Are you
Yeah,
tracking it?
Yeah, the one with that calculates your fat.
It does fat, it does muscle, it does bone.
No,
it
no,
does-
I don't have that. Mine's an older smart one.
Mhm.
It
Okay.
just does the fat and the weight.
Not as smart.
That
Yeah.
and okay. Yeah,
So
I am concerned about muscle mass because if you're not strength training, you're not actually I mean, what you're doing is excellent for your heart and your aerobic development, but for strength training, we actually need to tax our skeletal muscle.
Yeah, well, in the swimming pool, I've been doing a lot of air jumping and stuff like that because of I have neuropathy in my teeth.
Mhm.
Sorry, I'm making noise. Right? Sorry. Cooking my salmon. I have neuropathy, so I can't do a lot of jumping and stuff like that, and it also limits my walking. So,
Yeah.
but weight training, I do bands and stuff like that. with
Okay.
my arm.
Neuropathy. I mean, I'll leave it to Morley to say this, but
No, you can tell me now.
you know, Morley says all neuropathy is a copper deficiency.
Oh, wow.
I just haven't done it. I've been wanting to call him because I remember on his call with us, he said, just call me. I don't need to, I get concerned about copper toxicity and people, oh, you're taking.
Well, that's because they have sort of sold people on copper being toxic, but
So
the really good research and the copper experts and the animal nutrition, when you really dig into it, which is what we did in RCP training. I'm an RCPC.
Oh, I didn't know that.
Yeah. You see how, I mean, you know, as Morley says, missing information equals missing truth.
Yes,
And, you know, we are led to believe, and he spells believe with a capital LIE, the big lie about, you know, what the problems are. And back to the glyphosate issue, glyphosate chelates trace minerals,
yeah.
but it chelates copper, I want to say 100,000 times stronger than any other mineral. I may not have that exactly, but the order of magnitude of copper and then cobalt. So it's chelating these metals that are essential for, things like the production of B12 in the body. And really all the major enzymes and the functioning of the mitochondrial respiratory chain for energy production. So
So all the copper I'm taking, I'm eating very few vegetables these days because of my issues with my gallbladder and diarrhea. But I'm sure the fish and the meat have plenty of glycophosphates in them, right?
I mean, it really depends on your source.
Well, I don't have grass-fed available right. I do have hamburgers and a few roasts, but it's hard to come by a decent price.
Yeah.
Because I don't have a freezer. I'm considering that in order to buy more grass-fed beef.
Yeah, well, and even the grass-fed label, and when Alan comes and talks to us, we will learn a bit more about that because, truth in labeling, they allow a grass-fed label on cows that are grain finished.
Yeah.
So
Yeah.
you really have to ask, is it grass finished? Is it
Right. Yeah.
And I think about that too with some of the supplements that people take for, because they don't want to eat liver or organs or that sort of stuff. But so now there's this huge market for collagen, liver, organ supplements to supposedly get your nutrients that way. But I can pretty much guarantee you there's not enough grass finished animals to produce a high volume of those supplements. It's got to be coming from some of the cows who are not grass finished.
Probably.
And I mean, that's the problem with a lot of our labeling. So, I think Alan will talk about some of that. He's going to talk about, some of the fake food that they're coming out with in terms of, the lab grown meat. And I mean, it's, so I'm in a group with, a number of other CEOs from the supplement industry. And we have, the person who leads our group also is somebody who has worked in the supplement industry for years and, is now consulting for different people. And he runs this CEO group for us. And he was giving us an example last week of this great meeting he put on and, all the different things they did. And he mentioned that it was a company that's making this lab grown meat, and he was all excited about it. And I was like, oh,
How can anybody eat that stuff?
so anyway, I think Debbie, you had something you were going to say, but you might have forgotten it by now.
No. Actually, I've got 1+1 now. So you were talking about your beet kvass. So do you think the same thing might be applicable to sweet potatoes too? Because I had stopped eating because I read the same book that Mary did. And so I had stopped, sweet potatoes, and I had reduced my beets, but not completely because my husband loves and adores beets like crazy. So that the other thing too is that, so, we're out in Wyoming and my husband has found this supplier, and I don't know if they're from Montana or from Wyoming, but we get shipments of their beef all the time. And the owner, I think it's anyway, he writes all the time about like the cattle being out and all that stuff. And he had written an article about grass-fed, grass-finished, and how it wasn't as great as we thought it was. And so I should forward that to you. But what email are you using? Just the info at BiotiQuest or something else?
No, here, I'll put my email in here. I mean, so what I've read that too. Somebody else sent that to me. So in terms of tenderness of the beef,
No, this was, I think, more about the nutrients. I'll send it. I have to find it. And send it
yeah,
to you.
send it to me because that's going to really depend a lot on what they're being fed. So if they're feeding them a grain, but it's a grain that's got glyphosate, it's going to alter the cow's rumen, and that changes a lot of stuff. And it makes them fatter faster. So, I mean, you tell me if you think that. So,
And it depends on the cattle. It depends
Yeah.
on the cow, the kind of cow that they are grass feeding. Because if it's not a Charolais or a specific type of cow that does well on grass, and they're just trying to feed an Angus cow grass, I mean, it does depend on that.
Well, I think this was more about the pastures that they were actually going to and how the herds go up into the mountains and what they're getting versus just like a regular, I think, pasture. But I'll send it to you if you want to send me your email.
yeah,
then,
I put it in the chat there.
Oh, and then the third question is, so my friend's husband, they think he has Parkinson's, very early stages. So are you going to write a book or something that I can just give to people? Because it's hard, you know what I mean? Because they don't know, she has cancer. So for her to have cancer and then for him to be now getting Parkinson's, symptoms, it's a lot. So I'd like a little concise little thing to give to her so she can, you know what I mean? So, and then to have your success story and everything, I mean, that would be a really nice thing. And this would be an appropriate time with this data that you have now too.
Well, it's on the list of things to do, but it hasn't made it to the top. But I do have the Martha's Quest blog.
Okay.
So let's see, I'll put that in here. Where I write about Parkinson's. I mean, that's only writing about Parkinson's, alternative things for Parkinson's, resources. I talk about, I do try to do periodic updates on the microbiome, but so back to our oxalates for a moment. Gut microbiota and oxalate homeostasis. This perspective on how the gut microbiota can impact urinary oxalate excretion in the context of hyperoxaluria, a major risk factor in kidney stone disease. Constant elevation in urinary oxalate in these pH1, that's people with type 1, ultimately leads to tissue deposition of oxalate, renal failure, and death. And the only cure is a liver and kidney transplant, which is kind of interesting. But somewhere in here the most commonly described intestinal bacteria known to degrade oxalate are categorized into two groups: the generalist oxalotrose, including some strains of bifidobacterium and lactobacillus that degrade alternative carbon sources in addition to oxalate, and the specialist oxalotrophs like Oxalobacter formigenes. So I've heard about Oxalobacter formigenes, but lactobacillus and bifidobacterium.
Okay.
So I'd say open up, like make your beet kvass and put a thing of sugar shift in there. If that works for you.
Martha, I was going to ask you, I've been on your sugar shift pretty much almost a month before, I think Colleen got me onto it, before the challenge. And I have to tell you, I took 10 days off of Pilates and I went back hard. And the one thing I noticed that there was no muscle problems. Everything works so much better. But my actual question for you is I haven't slept since I gave birth to my first son. And I kept telling the doctor something has changed radically. And until I just listened to Supergut on a long trip. And it didn't talk about that, but when we were looking up his information, I saw his little blog on sleep, and then I see you have your sleep. You guys kind of don't have all the same ideas about which ones work. Can you tell me what you've found out?
For sleep?
Yeah.
Well, I'm going to back up to glyphosate again. I hate to
Okay.
keep doing it, but so there are 3 aromatic amino acids that we don't make, we get from our diet. And that is tryptophan, phenylalanine, and tyrosine. And tryptophan is very critical to sleep. And that the pathway that makes those amino acids in plants and in bacteria is called the shikimate pathway, which I talked about at the beginning.
Yeah.
So, and the shikimate pathway is the target of glyphosate. And what I would, this is why I believe with every part of my being that glyphosate is the primary driver of all of the health issues that we have in America, because the shikimate pathway is essential to all life on the planet. And so, our formula is working on the tryptophan. So it does have a little bit of tryptophan in it, but it's designed for the bacteria to be able to make that tryptophan again. And, we have our Lactobacillus plantarum that breaks down the glyphosate. So you shouldn't get interference with that path. And then our formula also makes bacterial melatonin. So the bacteria are melatonin producers as well.
Are you talking about
So-
sugar shift or the sleep
The
product?
sleep one. Although I will tell you, there is some bacterial melatonin production with sugar shift, a small.
Should I do them together or do I choose one?
We don't recommend doing them we recommend taking them separate, at least 2 hours apart from each other. And I'll tell you this is an anecdotal, but sugar shift is improving your energy production.
Mhm.
And the simple slumber is helping you wind down and sleep. And, they're designed as working teams, but if you put them together, you can, I'll say, accelerate the energy actually. And so I had a friend of mine who took them at the same time, even though I told her not to. And she was up all night with like this huge burst of energy. So, we design them as a team to do something together as a team. And when you start throwing something else into, or mixing 2 teams, you might not get the expected or the hoped for outcome.
So what I do, I don't know if the sleep, because I haven't ordered it. Is it a twice a day thing as well?
No, the sleep you just take an hour before bedtime.
Okay, so I can make that work because you do sugar shift twice, do that like sugar shift around dinner and then the sleep at night.
Yeah. And then,
Closer to bed.
it lasts. So that formula and the cross feeding lasts about 7 hours. So,
I'd take 7 hours.
typically by the time you wake up, and you won't have that sort of groggy other sleep aid type thing.
What are your thoughts on the, is it Yakult, Shirota?
So that's actually a really interesting, and that actually might benefit you some with sleep. So Shirota breaks down phenylalanine. So that organism is interesting. I actually looked at that and I've made yogurt with it. And I've had John taking it for a while because I think part of his issue is a problem with phenylalanine metabolism. So, and I even talked to some of the scientists at Yakult about doing a study in Parkinson's, because I think disrupted phenylalanine metabolism is a larger issue across the board.
Don't they test for that at birth though?
Well, not for those of us who are in our 60s. They do now. They didn't back then.
Oh, okay. I thought they,
And you can have
I guess you
like
can change too.
you can have partial dysfunction, or you can have an epigenetic change, which I would say is probably due to glyphosate exposure.
Right. Okay. That makes sense.
Can you tell me one more time, and I apologize, the three things that you say that we need, and one of them was tryptophan,
Tyrosine.
tyrosine.
Well,
you were-
tryptophan, tyrosine, and phenylalanine are the aromatic amino acids that are either made in plants or in grass that the animals eat, or made by bacteria in our guts.
So if I mean, I've tried tryptophan. I've tried everything, and I have had very little success. I think this
So-
makes sense.
You see these things here?
Yeah, your blue blockers.
Like, do you go out in the morning and watch the sunrise?
Oh my God, no, I can barely get up, but I can try. If I'm,
I
right?
think if you get up, like in 2 weeks, when you come back on here, if you've gotten up and watched the sunrise, I think you will notice a difference. So I have like a study group from my original RCP cohort that I was in, and we get together periodically. And one of the women in that group was she was having trouble with weight, and she was having trouble with sleep. And she could not believe, after just a week of watching the sunrise and just 15 minutes of morning sunlight, what a difference it made in shifting her. And so that's also why I started, putting these on. And these are my daytime ones. I have another pair that I don't see around here at the moment that are darker for the sun hasn't set yet, but it's getting ready to set. And when it does, I put the darker ones on. And what I noticed was, when I started wearing those darker glasses when I'm watching TV or on the computer, and I'm on the computer so much, I actually get very sleepy right after dark
When you wear those.
when I wear those. And
Okay.
I, try to limit, the artificial light exposure. And so the other thing is, the lights in your house, because they changed, you can't even get you can, there's a few places you can still get a fluorescent light bulb, but we went through changing the fluorescent light bulb. Changing the LED light bulbs, the blue light. Because this is also interesting. With John is highly, highly affected by these artificial lights. And he cannot go into a Costco store. If he walks to the entrance of Costco and tries to step across the threshold into those artificial lights, his entire body locks up. He can't move.
Wow.
And there's a book by a gentleman named John Ott, OTT, and I think it's called Health and Light. It's an old book. He's the father of photobiology, and he discovered by accident how different light frequencies change the male and female flowers of plants, and change the male female birth ratios of different animals, and studying this basically light as a signal. And then he went on to be kind of the guy who started filming light in cells using luciferase and these different tools that cause cells to fluoresce. So there's a lot to light. And yeah, I don't know. So when you said you haven't slept since you had your,
Sorry.
was it you said your first child?
Mhm.
Sorry. My husband just came home from his
Yes.
infusion. He has stage 4 colon cancer that he's beaten remarkably through this, but we were both on the gut. We just started the super gut today. It was as soon as I got home from the hospital, I knew something was wrong. I was so activated. For my
So did
first
you, did they give you antibiotics? Do you remember?
I'm sure they did. I mean, it's 25 years ago now, but I'm sure they did. I had an epidural. Yeah, I didn't have a
Yes.
C-section,
So,
so
I mean, uh, the microbiome is just so essential to who we are and to maintaining all of these hormone and chemical balances in the body. I mean, this almost 10-year journey with founding the BioCollective and learning what microbes do. I mean, I'm amazed we have gotten this far and done this much damage without somebody going, hey, wait, we're killing ourselves.
I agree.
Mhm.
I think you're on to I think this will hopefully, if you're not blocked by, big corporations who have everything to lose by this, hopefully this will be the new medicine.
Well, I sure hope so. I mean, I just want to help people, and so there's also I don't know if this would work for her, but this is a little biohack that I think I might have mentioned this on the book club, but this scientist who's helping me with my Parkinson's theory. So there's a number of bad bacteria that we know in Parkinson's that are specifically implicated. And some of those produce an enzyme called urease. And the urease will kill the good bacteria. And when it does that, the good bacteria spill out all their potassium and calcium, and they make this colloidal mass, which is a very rock hard stool. So you get an electrolyte imbalance, you get a stool that's very difficult to pass and has a lot of toxins in it. And you get this concrete like constipation that people often see in Parkinson's. And the bacteria that make that urease are temperature sensitive in terms of below 37C or 98 point whatever, the human physiologic body temperature. At physiologic temperature it will make the urease, but if you drop the temperature down it will not make the urease. And so we have these little ice packs, it's about the size of your belly, and John puts the ice pack on when he is eating, or he puts it on, he also puts it on when he goes to bed. And so he sleeps with this little ice pack on his belly. And we've definitely noticed a difference. But you have to be vigilant, because we notice if we slack off for a day or two, the problem comes back.
So if somebody never runs a temperature of 98.6, but always lower, we wouldn't have an issue with it, right? My temperature
That's actually not true, because it's a local temperature. And so John's forehead temperature is typically 2, sometimes 3 degrees below normal. So I think of that as like maybe his body's trying to do something, but it's not having the effect in the local situation where it needs to have an effect.
Interesting.
I mean, the body's pretty amazing.
Yeah.
I had that when I had Rocky Mountain spotted fever and it was undiagnosed. I had this massive body rash, and when my body was cool or colder, I wouldn't itch for like like I would itch all night long if I got under the covers and got just a little warm, like normal body
Right.
temperature. I would itch so badly. But when I was cold, it was actually, it's kind of interesting, the body trying to regulate so
Yeah.
that, mhm.
Yeah, wow. Mosquito bites don't itch when you're cold either.
Thank you so much. I have to get up, but thank you so much. We'll see you in a
Well,
couple weeks.
it's time for us to wrap it up anyway. So, are there any other questions I can answer real quick, or
What are your
if not-
thoughts about the microbiome feeding when if you are more carnivore than not?
Ben Azadi actually has some recent content on that specifically. He did a before and after microbiome test and actually showed that a lot of the keystone species actually came back when he was doing carnivore. And I think what people miss, of course, he was probably doing carnivore in a much, I'll say, a much more pure way than some people would do it. Because Ben teaches it. So he's doing organ meats and different types of cuts. And, he's probably not just eating a steak every day.
Right.
But carnivore diet is high in hyaluronic acid. And that is a fiber, and it is the fiber that is used to make the tissues in our body, collagen. And I think I actually read in a book I have where I was working on this, the article for Weston Price, that at some point in its life cycle, every cell in the body has the capability of making collagen.
Oh, wow. We
Wow.
wouldn't take collagen in our coffee.
Yeah. Well,
Okay. That's great. Okay, thank
so
you. So what is
anyway.
Ben Azadi's group name? I remember him, but I can't remember the name of it.
Well, he's got a new name. What's the new name, Jen? Do you know? It was Keto Camp, but the new
Right.
podcast is Metabolic Freedom. I
Metabolic
think.
Freedom. I think that's the title of his new book coming out. And I put a YouTube link where he talks about the carnivore experiment. And I just, I do want to kind of caution people though that, it makes a difference whether you jump into carnivore with a healthy gut versus using a
unhealthy.
strategy to, like get over SIBO. I don't see that working particularly well. It can help with symptoms for a while, but I don't think it's a long-term strategy. But doing it when you already have a nice, healthy gut and then jumping into carnivore, I think you can have just really incredible results. So timing and health, other nuances kind of matter.
Yeah. Yeah.
I would agree with that, and I would, I just question extreme diets in the long term, whether that, we weren't really made for that. I think most of the problems we have with our gut and our inability to eat these foods is, the chemicals that we have put into our food supply and the processing and the preservatives and, all of these different sort of pieces of the puzzle that are killing our microbial self.
Yeah.
So with that happy note, we're here to restore your gut. And so thank you for being part of the call, part of the challenge. And, if you have anybody else who wants to join, I did put my email in the link. So if anybody's still having trouble signing up, or if you know somebody who wants to participate and they're not having success signing up, just have them email me and I will
Yeah.
get to the bottom of it.
And Martha, I get a lot of questions from people that if they're already part of Sugar Shift, are they getting the emails for the book club and vice versa? Because people want to be included in both, or are they separate?
Well, it's two different lists because we had people complaining about, I don't want to be in the book club. So we separated them. But what we can do is, we can send out a reminder to our kind of the whole list of people, if you want to be on the Sugar Shift calls, even if you're already doing Sugar Shift, you can join those calls, and here's the link for that. And if you want to be on the book club, here's the book club calls, and let us know if you want to be on the reminder list.
Okay, great.
And then also, Jen has done a survey. We're going to be sending out a survey to people to really just kind of get some feedback on what are your primary goals? What are you looking for out of this challenge, and how we can best support you. So we'll be sending that out over the next week as well.
Perfect.
Great. Thanks, everybody.
Thank you, guys.
All right. Bye-bye.